This protocol dissects the individual mechanical properties of the cell nucleus that make it a mechanical gauge for cell polarization, differentiation, and migration.
The main advantage of this technique is that it can measure mechanical properties and apply forces to subcellular compartments without external perturbation of the membrane or cortical cytoskeleton.
The protocol can be easily adapted to manipulate beads inside other cells of other organisms in order to study nuclear mechanics in different cell lines or animal models.
Alignment of the direct force sensor is critical to capture all the light creating and leaving the optical traps. This will ensure accurate light momentum measurements in viscoelastic medium of a cell.
Begin the single-cell preparation by placing the sphere-stage embryos, 4 hours post-fertilization, in a glass dish containing E3 media using a plastic Pasteur pipette. Then, select the embryos that are positive to the beads and express the fluorescent protein in case of mRNA injection. Use forceps to manually decorionate the embryos and transfer approximately 10 to 15 decorionated embryos to a 1.5 milliliter reaction container with the help of a glass Pasteur pipette. Remove the E3 media from the tube and add 500 microliters of pre-warmed carbon dioxide-independent tissue culture medium. Gently shake the tube, avoiding bubble formation, and ensure that the contents of the tube become turbid as the cells dissociate, with no big chunks visible by the eye. Then, centrifuge the tube at 200 times G for 3 minutes. After centrifugation,